My name is Pinzu, and I'm from the Institute of Biophysics of the Chinese Academy of Sciences. My research is working on the cryo-electron microscopy and cryo-EM method in structural biology. So today I will give we have two lectures. The first one, I will give a brief introduction of how cryo-EM works, the principle, and how it works. And the after break, I will give you some examples, mostly in my lab, what we are doing using cryo-EM to get the structure of virus and molecules. So the first one, I will get started of the cryo-EM. Actually, in my institute, and like Professor James Liu, we are working on mostly on a lot of the structure biology. So this one is give a lot of methods about how we study the structure and the functional study of the molecules and the complexes. Basically, so the most important method and the most popular method in the structural biology is X-ray protein crystallography. And I think this tomorrow, Professor Liu will give the introduction about this method. So today, and I will focus on this one, the cryo-EM electron microscopy method. So basically, so this is one of the methods. So basically, so in the structural biology method, there is what we call the PDB. That's what the structure solved. How many structure solved we deposit the coordinates into the database. So this is you can see the trend. So listening, so you get increasing. So the structures deposit by the whole world, the structure biology but you can see the tendency. So it getting more and more, and getting more. It should be stable in this recent years. But if you look at the contributed by the EM field, so basically from the first structure deposited by EM field is around 1980s. But now you can see it getting more and more and more, especially recent years. You can see there is a breakthrough of the how many structures solved by the cryo-EM field. So this is my personal statistics. 2 days. This is when we start trying to get apply for a proposal in China, and at that date we found there are about 240 EM structures in total deposited into the PDB database. But now you can see as of today, just to see there are more than 700, and even for this year alone, there are more than 200 EM structures. You can see the tendency, so getting more and more popular in the EM field. So basically, the cryo-EM, how we do the cryo-EM study. So basically, we don't need a lot of sample. We just apply a tiny little amount of the samples, like 3 or 4 microliters, apply to our what we call EM grid. Then we use a filter paper to blot it, to make it a very thin layer of the sample suspended in the water. And then we drop into the liquid acid to make it cool down. The cool down is getting cold very fast, like more than 20,000 degree per second. So getting very fast. Then we got some of the EM field. So we got the sample suspended in the vitreous ice, what we call glassy ice, because the speed, the cooling speed is it's too fast to get in the ice, the water getting crystallized. So that's what we got the EM grid inserted into electron microscope, and this one. I will show you later how we do the 3D reconstruction. So this is basically the idea of the cryo-EM, how cryo-EM works. So why do we use electrons? Because from the principle of the Broglie. So the limit you can tell of the light is a half length of the light you use. So basically, if you use visible light, the the visible light, the wavelength of the visible light is around 400 and 600 nanometers. So that means the resolution you can tell is half of them is around 200 nanometers. That was the principle of the light microscopy. So, but for the electrons, the wavelength is much more shorter. So that means the limit of your resolution is much higher than the visible light. So that's the reason we use electrons. So give you a much more further detailed information of the structure. So basically, this is the older electron microscope. So that's electrons getting out from here. Then this was transmission electron microscope. How we use this one? So basically, there we like light microscopy, we have a lens. But here the lens was made of electron and magnetic. So basically, make the electron beams focused and spread and getting higher. So this is where get a projection. So this is the final screen you will see your sample. So that's what electron source we use now. Getting better and better of the electron microscopes getting in the market. Basically, for example, electron source we got what we call field in Michigan. So get very highly parallel electron beams give you the very correlative electron beams, give you the higher resolution and the focusing, and a lot of detectors. I will get in this later. For example, we got previously we just used negative films, and we have to develop it and scan it to get it, digitalize it. It is very time consuming. Now we got a CCD, it's a digital camera. In recent, especially this year, there is we call a new device we call a direct electron detector that we can count individual electrons. This one gives us very. There is about 2 weeks ago, there's a breakthrough in the EM field. I will show this later. So we using cryo-EM to solve not in my lab in this field. Solve the ion channel in the 3 angstrom resolution. That's very a breakthrough. We don't need a crystallize of the membrane protein. We can see it very clearly. So this published 2 weeks ago in Nature, 2 articles. So this is the principle of TEM. Now, because so how can we see that? Because a lot of our biological samples are made of carbon, oxygen, and nitrogen. Of these atoms, is very low number of the atomic number. So how can we see that? So basically, we have 2 kinds of the method that we can see. One is called a standing EM, and another is what we call cryo-EM. So for the standing, so to increase the contrast, because of our as we said, biomolecules are all the carbon, nitrogen, oxygen, these things. So it's hard to see. So basically, we use heavy atoms interest to enhance the contrast. So, but there are several ways to do that. Positive standing, just you incubate your sample with the heavy metals. And what we mostly use, we call it with negative standing. That basically we sprayed a layer of the heavy atoms like mostly is tungstate or other things. So in the background, then you can see the background is heavy, is black, but the molecules, because the heavy atoms is not get through, so you can see the white. So basically that's what we call a negative standing. Another thing is also sometimes we make a shadowing heavy metal on the surface, so give you a contrast. So this is a typical negative stain image of the virus. This is I'm not sure. This is a tobacco mosaic virus. It's a long, so 2 typical virus. So use this one. The advantage of the negative stain, you can see the high contrast, but disadvantage of this one is the size of the staining will reduce the resolution. So actually, you can see just the morphology of your sample, but not much more details. In the contrast, so cryo-EM, so we avoid the harsh staining. So we don't use staining. we just apply the sample directly into the sample. So we freeze it, just as I showed before. So for this one, the advantage of this cryo-EM is the sample structure unchanged. So basically, we can use the buffer most suitable for your sample. We can keep it in the native status of the molecules and freeze it. And so all of the inner structure of molecules is accessible. So in this method, this is what the advantage of the cryo-EM against the regular negative stain EM. So this is two examples actually made by myself. So this is HIV AIDS virus. So this is a negative staining virus. So you can see a lot of contrast. Basically, what we are interested in is the surface protein. I will in the second seminar, I will show you why we care about the envelope spike on the surface of the AIDS virus. So this is negative stain. You can see a lot of contrast. This is a cryo-EM. You can see what we also, if you also interested in the surface protein, you cannot see. It's hard to see. But the things for negative stain, it will disrupt your virus. So structure getting, so the whole virus structure is getting disturbed. But this one you can keep it in the good shape. So this is a good contrast, fine detail, but distortion. But this one is distortion free, but this is low contrast. So we have to develop some method to enhance the contrast, and so you can see it more clearly. That's what negative stain EM and cryo-EM. So this is two examples. So basically, this is how we do the cryo-EM, the whole pipeline. The first thing, of course, you have to make homogeneous sample. So this is a wet lab story. So if you are interested in the virus, you have to purify the virus first. And if you are interested in some molecules, you have to, for E. coli, insect cell, or every means. The idea is like protein size. So you have to get a very homogeneous sample. So this is a biochemical preparation. So after you got the homogeneous sample, then what we do is then you freeze that. That's what we used before. We showed before. Then you get the cryo-EM sample preparation. Basically, that's apply 3 or 4 microliter sample into an EM grid and freeze it. Now we get a good device to reproducibly freeze the sample. After get the EM sample, insert into electron microscope. So this is in the EM. How you see in the electron microscope to see how it look like. This is the virus sample. So you basically collect a lot of this kind of images, thousands, maybe thousands or 10,000 images. Then we have we have to process the data using the image processing. Basically, then a lot of different software are involved here. Then you will get a 3D density map of your sample. This is a typical virus 3D EM density map. So it depends on how the resolution. Usually, for 3 or 4 years ago, the regular resolution we can achieve using cryo-EM method is around maybe 10 angstrom, 1 nanometer resolution range. In this range, you cannot see very detailed things of the atoms, like side chain of your amino acid. But the good things for the cryo-EM compared with X-ray things, you can, for example, the whole virus is difficult to crystallize, but you can get the whole morphology, so how it's overall organized of your sample. Then in each subunit, for example, in this here, in each subunit, if you have some high resolution structure getting from NMR or X-ray studies, then you can try to dock the high resolution structure into our low resolution EM map. Then you can get a pseudo atomic model of the whole different subunit, the whole morphology of this. This is a virus, sometimes a complex protein complex. So you can see how this this subunit are interaction to each other. This is a typical way of the EM study. But recently, there is a lot of development in the cryo-EM field. The resolution of the EM reconstruction getting higher and higher. If you get the 3 or 4 angstrom resolution area, so this is what we actually in my lab or in other field also get other labs in this field also achieve this kind of resolution for some typical axial virus like this one. So if you get into the 3 or 4 angstrom resolution, then you can develop the whole atomic model of the virus without knowing the X-ray structures. So you can build from the novel. So this is depends on how you achieve the resolution. So you can build a real atomic model or pseudo atomic model. Then you can do this is a typical way of the structural biology. You have to do some structure analysis and do some functional assay to test your model is right. So this is the pipeline of the cryo-EM study. So from the sample preparation to getting the final structure and some functional study. So this is an overview of the. So there are several ways in cryo-EM field. We call and depends on what sample you can get. There are 3 ways basically. It's single particle. Single particle. I will talk a little bit. This is the most popular technology in EM field and the electron tomography. And also sometimes we can get the 2D crystals like 3D crystals in X-ray crystallography. In 2D crystals, basically, so it's very highly periodic. So you can use this one to the electron diffraction. This one can achieve very high resolution, atomic resolution, for example, 2.5 angstrom. So this is comparable with X-ray. But the things for this one is also, so there are typical some membrane proteins. There are several membrane proteins are solved in this way, but it's kind of comparable. Is even more difficult than the 3D crystallization. So if you want to get a 2D crystal, it's difficult to get. So basically, there are other things is a single particle and the electron tomography. So it depends on your sample. If you get a homogeneous sample, you can get using a single particle. And this one, you can even can go to the electron cellular cell biology things. So you can do a 3D reconstruction of your in the cell background. So this is the idea how we get the 3D reconstruction method. Basically, we just from now on just focus on the single particle and the electron tomography. So basically, the idea is like in the CAT scan in the hospital, if you want to get the 3D shape of for example, brain, some disease. So if you, this is CAT scan, so basically the detector getting all around the object you are interested in, the different projections. You get the projection image from different angles. So this is what CAT scan as a principle. So in the electron microscopy area, the electrons cannot tilt. So basically, the electron always getting through from top to bottom. So so alternatively, we can tilt our sample. So this is you can tilt your sample in different projections. So basically, you got a projection image from different angles. Basically, you get a 2D projection image from different angles. After that, we have some principles to do the 3D reconstruction. So based on this projection image, you get a reconstruct your sample. So this is what we call electron tomography. This idea is you focus on one object, you get a projection image from different angles. This is and another way is we call it single particle. Basically, we just shoot a lot of pictures of the objects. So you always get a 2D projection of your 3D project. So if we treat these different 2D projections as a projection of one single particles in different angles. Then you can almost the same thing. So idea for this one is basically we got a single particle image of the projection, but we treat it as look like this the objects are tilting, but it's not real tilting. So the idea is that so you got different projections. You treat them as a different projection of the one single object. So the requirement of this method is that your object must be very homogeneous, but they just in different random orientation. So this is the idea of the single particle. So basically we have two kind of things. So for the single particle analysis, the basic requirements that your sample, your particles must be homogeneous. So I will give more details of how single particle analysis is working. So basically, this the same idea. There's an old method, all the way we brought the sample like we showed before. Then we collect the images. So this is what this kind of project will get the 2D projections. You can see this orientation, or this orientation. This is kind of tilt. This orientation, or this. This some kind of top views, so to this way. So you can get this side views, tilt views, and top views. So basically, you got different orientations. Then we just pick these particles, a lot of particles, just get them averaged to enhance the signal to noise ratio, and we treat them different cluster average as different projection images. Then we do the reconstruction. The goal is to get the 3D information of the project. So this is overview. So what we use the EM sample grid. That's what we apply the sample in here. So basically we coat the EM grid with a thin layer of carbon, and on this carbon there are kind of holes, different size, small holes and big holes. Then, because in these holes, there are a thin layer of your sample, so you get some lens ice here. So this is what we look like. This is the old EM grid, but now we get enhanced. We get a very regularly placed holes on the surface. So this is what we got the EM grid. So this is the blotting process. I will skip on this one. We just apply this sample here and use a filter paper, blot it, and plunge it down to get it cooled down. So if we got this, you get it right, you must get a very thin layer of your sample. Then what we got is this kind of ice. We call it vitrified ice. It is like a glassy ice. There is no crystals. So if you using a diffraction mode, there is no period, no dots. But if the ice is too thick, or the speed of the cooling down is not fast enough, you will get this kind of crystallized ice. So this is not good to our EM study. So we want to use this kind of ice. So basically, you have to get a very good thin layer of the ice. So basically, this is what we want. And then after we got this the EM grid in here, we transfer it. This is a sample holder, which will be inserted into the electron microscope. This is electron beam where electron beams getting done. So this is where is a EM grid getting here. So in the whole process we keep it cool by filling the liquid nitrogen here to make the whole sample holder cold enough, around minus 140 degrees. So then the electron beam is getting. So then in the whole field you have to be very careful. Don't make it warm up. So make the sample keep it frozen. So this is a device what we use, the cryo holder. But in the newly designed electron microscope, we even don't need this one. We can use other devices, and you can send up to 12 EM grids at one time into the scope. So it's much more convenient right now. So this is then we after insert the sample in the electron microscope, then we got the TM image. We collect the data. So this is a pseudo-colored EM image. So this is a typical image of the virus sample. So basically, for this one, you can shoot a picture and then go to another place, shoot another picture. And previously, we just used negative films. Now we use CCD camera to do this one. So we so this we showed before this. Now we have a software which can automatically collect the data. You can find an area, go to another place, and then shoot another picture. So keep going on. You can remote control these scenes. So you just basically got a lot of images of your sample. Then this is the principle of the image processing. So basically, this is a 3D project. We got the 2D projections. You want so this is, if this is side view, from this view you can feel what your projection look like. But this one maybe is difficult. It's kind of difficult. But the idea is that you want to get all different orientations, projections. So then you trying to get the 3D object back. So for here, it's easy to imagine. But for this one, it's very difficult. From this to the projection, you can never imagine that this is this kind of 3D objects. So this is so idea is that you want to get the different orientations. So this is our plot to see the orientation. So basically, you want all the dots distributed evenly around the full space. So now that means you can get all different projections from different angles. So, but sometimes it's difficult. For example, some samples, they tend to lie down. For example, if this is a rod shape, this one, they tend to lay down this way. It's hard to make them in this way. So this one, we have to make some trick things to make them random orientated. So this make the sample. So this is a whole overview of the how we do the single particle analysis. Basically, you got the cryo-EM cryo-EM pictures. Then you box the particles out. We got a lot of particles. We call single particles. Then we trying to align them together, trying to get to the class average. So this class average, then for example, you have 100,000 particles. Then you align them into 100 classes. Typically, in each class, you around average about 1,000 particles. 1,000 particles, you get much higher. If you get them average, then you get a much higher signal-to-noise ratio. You see the contrast much clearer. Now you get 100 class averages. You treat them into 100 different projections. Then you got 100 projections down. Now you can get a guess. You can guess a 3D shape of this one. It's for the first several cycles, the 3D shape may be not right. So you can try to reproject them, refine your structure. So getting a coarse 3D model, and now finally get the final 3D map. So this is an overview of the single particles. How do we do the image processing? So basically, do the classification and do the initial 3D construction, then do the refinement, finally get your final model of the 3D shape. So this is how the single particle work. So I will show some examples in the next lecture about in my lab. So another typical ways is electron tomography. So the principle we have already showed. So this electron tomography is the objective, and we can treat is different. For single particle, as we said, we have to get very homogeneous. You have to purify your sample out, get a homogeneous sample. Why we want to do tomography? Sometimes we want to see the 3D shape of your the interesting objects, for example, molecules or other things, but in the context of your cellular background. So we want to see your sample, how it look like in vivo, for example. So basically, that's what sometimes we want to the electron tomography. So this is is needed to resolve the factor in this range. So it's good since it can go to even for the cellular background. So the method of the electron tomography of these ones is basically we are not doing the single particle. We just focus on one really single particle, just one particle or one object. We just tilt them, as we said. We tilt them in different angles. We get a 2D projection of the same project in different projections. So because of the limitation, basically actually in cryo-EM, the number of the projections is limited because you cannot do. You have to use very low dose of the electron dose because this will damage your sample. Especially you want to take a lot of pictures into one single object. So basically, and also the number of the projections is limited for around, for example, 100 projections. So so we cannot fully cover the whole projection. So we have if we take this 3D to 2D, we use this one as an example from 2D to 1D. So this is 2D image. You just project in this way. So you get the 1D projection image of this one. Then you use this one. You can try to okay. I'm sorry. We should maybe. Now after get this one projection, then we use 2D projections to reconstruct the 3D projection object. So this we still use from 1D to 2D projection. So this is you get a 1D projection of here. You can get a 1D projection here on a different maybe this one. So you just use this 1D projection. If you fully cover the whole projection orientation, you can cover this image. So this is the reconstruct 2D image from different 1D projection images getting this one. But if you compare the original pictures, you can see some distortion because you cannot fully cover them because you always the sampling there are some missing information in area. So basically, what we can do a dual set or other things. So it's basically that's the idea of the electron tomography. So in practice, what we can do is that we just insert the sample here, this stage sample stage can be tilted, and it can be computer controlled tilt because you can imagine because we get very high magnification of the EM field. So if you tilt, you can imagine that you just tilt a little bit, but because a big magnification, if you lost, you can easily lose your image because of very big magnification. So you have to very good control, well controlled the tilting. If you tilt a little bit, and if the sample is here, but it will go to that way, so you have to bring it back. For the cryo-EM, it's very difficult because you cannot shoot a lot of pictures. You have to save the dose for your really imaging conditions. So you have to use that's what we have to develop some software to make it tilt it and make it back and shoot a picture. So that's the reason the principle of electron tomography has been developed about more than 30 years ago. But the really application of this kind of technology has been useful is just recently, maybe 5 to 7 years or so, because a lot of development in this software control and also the development of the EM device. So this is what that's why I said why we have some missing value, because we cannot fully cover the area. So you cannot tilt to this way minus, because as I said, there's a mechanical limitation. You cannot tilt to minus 90 degrees. You can almost get into highest minus and plus 70 degrees. So that we have some missing value because you lose some information in this area. So that's what if we can tilt or cover all of them minus plus 90 degrees, so you can almost recover the image. But if you only use the minus plus 60 degree projection, you can cover. So that's a distortion of this one. You can always do another thing. You can do what we call the dual axis tilting. So that means you tilt this way minus 70, and then you rotate your sample. So you tilt this way. So the axis tilt this way. So you can cover more information still. Dual axis you can cover more. So that's what we use dual axis. So trying to cover as much as possible information. So basically that's electron tomography. A lot of application of electron tomography is a cellular biology. So basically, because the cell is too big to be observed in EM, then we have to cut them into thin layer, very ultra thin sections. So then make the electron beam can get through. So that was so basically conventionally in the typical EM lab. We use the chemical fixation, dehydration, and trying to embed them into plastic and trying to cut them into a very thin layer of the plastic. Then you stain it. So this is what we call the conventional. Now we can do the cryo section. So basically freeze it and cut them in cryo conditions to make the structures native. So this is how we make the samples conventionally: freeze substitution and cryo section. I will show a picture of later on. So this basically, so what we do, we make the sample of the thin section sample and you do the electron tomography. So basically, that's what there are some examples of the electron tomography. This is a very typical one. So this is how get the cellular image in the 3D. So this is segmented and colored. So you can see the molecules or other things very clearly here. You're getting a higher contrast. And another thing, instead of the cells, you also some samples, for example, virus. I will give an example of HIV virus or some herpes virus. This one you can use in a single particle, but the size of the whole envelope virus, the size is even different size. So you have to focus on the one single virus using electron tomography to do the 3D reconstruction. So this is another example of the electron tomography using deal with the virus. So there are several examples. So I just showed several principles of the single particle and electron tomography, which are mostly used in the cryo-EM field. So in the end of this session, I will give you some challenge and the current progress of the cryo-EM field. So basically, the challenge on the development of in this field, they are focused on several things. First one, we want to improve the resolution. So basically, we want to get the atomic structure without crystal. So this we are getting more and more structure in getting into this area. And also, we want to try to combine EM and other methods. For example, if you just show, we get a low resolution EM map. You want to combine the high resolution crystal structure and form a morphology of big complex. Also, you want to visualizing a biological process using electron tomography of the whole cell things. And this one, all of these things are very time consuming. So a lot of label and a lot of things we want to do the high throughput, do it fastly and automatically. So this is several things in the cryo-EM field. I think trying to getting things give you. So first thing is you want to get the high resolution. So this is typically maybe 10 years ago, you get this kind of EM field, EM map. But now if you getting more higher and higher resolution, if you go to this range, for example, 4 angstrom range, at this range, you can try to make the atomic model of this one. So this is an example. This is a virus. This is a virus of the, you get it about in 2010 in Professor Hong Zhou's lab. They built the full atomic model of this virus. You can see this side chains very clearly. So this is basically axial virus. For this virus, you can take advantage of the symmetry, axial symmetry. So this one you need much less particles to get a higher resolution. So this is a typical things in EM field. For the axial virus, you can get a high resolution, atomic resolution. Even in my lab, we also get some high resolution, atomic resolution virus structure. And since for this one, this is I mentioned before, just 2 weeks ago. This is EM channel. EM channel. This is a very challenge objects, and very difficult to get a crystallized membrane protein in the EM channel. So this is without axial symmetry. For this one, you can see now so this we think is a breakthrough work. So this one you can get they got a 3.4 angstrom resolution of this EM channel membrane protein without crystallized. So this means that this cryo-EM field, I think we are getting more and more attention because we can deal with this kind of difficult challenge objects. This is a good development of this one. So you compare with one. So this is the first, maybe the first near atomic resolution EM reconstruction in 2008. And this is a crystal structure of the same virus, the same field, the same area. So you can see this alpha helix is resolved well. So now it's getting more and more structure solved near atomic resolution area. The second trend of is trying to combine different things. You can integrate with bioinformatics. For example, as I said, we want if you know the atomic resolution, atomic structure of the subunit, but sometimes even the subunit is not crystallized. Then we can try to combine with bioinformatics with the constraint of your EM map, because then they can try to predict the structure, but with without any constraint. So the prediction, I think, is not very reliable. But after you put some constraint, for example, with the EM field, EM density map as a constraint, so the prediction is getting much more reliable. And so that means the same thing. You can try to modeling with cryo-EM restrictions. And also you can integrate with different data type. So this is typical. Ribosome is another sample getting used in the cryo-EM research. So you can try to model this one, EFG. This I'm not very familiar with, but this is an idea of that one. So if you get in, that's what only 10 or maybe even worse than 10 angstrom resolution, you can use that rigid fit model. But after you achieve to 5 to 6 angstrom, you can see the alpha helix. Now you can try to use the density map to do the modeling. So this with the help of the EM density map to do the modeling. And if you get into to the higher resolution, for example, 3 to 4 angstrom, this is my work, the work in my lab. So this one, now you can build the atomic model without any help from the crystals. You can see the side chains to build the whole atomic structure. So this the third chain of the we want to try to do the electron cryo-ET of the whole cell. This is another trend. So basically, this is what this is the area of cell biology. We use light microscopy. You can see the, for example, confocal. You can see the whole cells. And this is what we use NMR or crystals or proteins. There are always a gap between them. So you see the high resolution structure, atomic structure, but how it looks like in the cell context. So basically, the electron tomography can bridge the gap between this high resolution and low resolution work. So that's what we call it with structural cell biology. Using electron tomography can play an important role in this area. So for example, if we are, actually my lab also interested in to visualize the virus cell interaction, how virus bind to the receptor, how to get into the cell. So this one, we can try to use electron tomography to visualize the process. So basically, in this one, you can use we call it correlative microscopy. We use both light microscopy and electron tomography and ET. So basically, that's the idea. So you get a sample of your cryo-EM sample, but here you have a tag tagged to the proteins you are interested in. Then you put this one into the light microscope. You can try to localize the proteins with the tag, with the help of the tag, localize the position of your proteins. Then you transfer this EM grid to the EM electron microscope to get more details, and trying to with the help of this localization, in trying to find the pro molecules and using electron tomography to get a 3D cancer. That's the prototype and idea of the correlative light and electron microscopy. So basically, in our institute, we are the director of our institute trying to lead a project to build a platform to try to combine the EM and LM. Actually, they use a high super resolution LM. I would call it IPAM. We use trying to combine the IPAM and the TEM, or IPAM is SEM, scanning electron microscope, to try to do the correlative things. So basically, that's what cryosection. After you get a cryosection, you get the light microscopy and the SEM and the TEM, trying to combine them together. This is how it looks like. So basically, we trying to freeze the sample, then get to the actual thin section, put them in EM grids. Then first thing you trying to visualize them in the light microscopy, trying to localize it. Then put it into EM. This is EM grid. Now this is computer controlled. You can electron tomography tilt them in different angles. Then you get the different angles projection image, trying to do the 3D reconstruction, and trying to localize. This is a prototype, trying to localize your molecules. Then focus on this one. You can try to get the 3D reconstruction in the context of the cell. Another thing is we can try to combine with LM, that means light microscopy, and SEM. So how can we do that? Is that basically SEM is basically scanning. You get the surface feature of this one. So after you get the 3D things, you can try to embed your cells into make a plastic things. Then you try to in the SEM, you can just cut them in one slice image, cut another slice, get an image. You can you can go through these ways, so you get a full in the big area. You can get a 3D construction. This is we call it a 3-view. That's a mechanical section stuff, and also sometimes we use we have a dual beam SEM. That means electron beams you can do the imaging, but there is another ion beams can do mirroring. So basically, you don't need the mechanical knife to cut them out. Use another beam or the ion beam to cut them. You can cut a very, very thin layer out. So basically, you can improve the resolution. So idea is that we really want to. This is a courtesy of my friends. They supplied this movie to me. So we want to see the whole scenes of what happened into the cell. You can see clearly, trying to using electron tomography to see how. This is what he is interested in, the molecular motor, how it distributed and around it in the cell scenes. So basically, that's it. And this time consuming, we want very. This is several chains in the field. So for this one, Protein device, this manually. I used this before, but now we call it virtual board. Basically, we can do it very highly reproducible, get a good freeze sample. And this is a cryo holder. Now we got a new scope. We don't need this one. You can transfer up to 12 grid into the scope. Now this is holy grid. Now we developed a new grid. It's very regularly holes here. So do this one, you can do the automation. After you collect image in this hole, you can just go this way hole. Yeah, just do it one by one. Now you can collect the images automatically. And this year's the most important development is go to from CCD, the direct detector, go to the direct detector. That's what I showed you before. That's why they got the membrane protein solved. So basically, in this whole process, we're trying to get the automatic particle selection, automatic structure determination, and automatic discovery. This build as a whole platform of cryo-EM field, make it easy and fast. So basically, the summary for this session is that, give you the whole message is that for the cryo-EMS, no crystal needed, and it can preserve the 3D structure of the sample. And you need a lot of particles to do the reconstruction. And also, the most important thing is a lot of people and the collaborators ask us. So the sample for the cryo-EM can deal with is around 300 kD. If it's smaller than 300 kD, it's kind of difficult, but this field is trying to push the limit. Maybe 200 kD, we can give it a try, but less than that one is difficult. So, but for the big complex, it's no problem. The problem is that you need to get a homogeneous sample. So this typical resolution is 5 to 20 angstrom. So this is a cryo-EM field. But now for some special samples, for example, axial virus, we can achieve near atomic resolution, maybe 3.5 or something like that. So this can be feasible. Actually, it's already feasible, but we're trying to get more and more samples to get into this area. So this is I recommend to the students in our institute to give some text and the web page of the how cryo-EM field. This is some materials. And the take-home message is that I just use the slide. I just give this class a lecture in our structural biology class. So hope the student can understand the structural biology. So what's the advantage and the disadvantage of the cryo-EM vs crystallography, and what's the principle of the single particle and the electron tomography, and how about the resolution as a development. So I think maybe we can give a maybe 5 minutes break. For the second session of my talk lecture, we'll give some examples, take advantage of the principle of what I introduced in the first half of this section. So basically, I will give you some example both for the single particle and the electron tomography, since mostly in my lab or some work I work in US. So I have already showed this one. This is the whole overview of the cryo-EM study. And the cryo-EM, there are several generations of the TEM. So basically in IBP, we have this generation, and the most advanced one, what we call Titan Cryos. So this one, you don't need the cryo holder, like I showed before. Then we can supply up to 12 cryo-EM grids. So this way we have the Titan Cryos, and this is this facility in our institute. Titan Cryos, some high pressure freezing, the freeze substitute for the sample preparation. And after we collect a lot of images, we have to do the high computing image processing. We have a high performance computing stuff. Now it's supported by a previous postdoc in Professor Liu's lab. Yeah. So basically, this we have both single particle analysis and electron tomography. That's what we have already mentioned. That we have two kinds of technologies. So in my lab, we use both technologies, both electron tomography and single particle analysis. By the questions in my lab, we are interested in the two questions. One is virus related, virus infection and replication. So the infection is how it get infected things and get vaccines, and how virus get replicated. Another topic we are interested in actually is epigenetics related. Is how about the chromatin structure and the chromatin remodeling complex structure. So basically, we have two kind of we are addressing two kind of questions. Is how virus get infected, and the virus get replicated. And another thing is how chromatin is organized and how it's regulated. So I will give you, for the first thing, an example of the electron tomography of the HIV envelope spike. This is not not the work in my the work before I went back to IBP in China. It's work in US work when I was a postdoc out there. So this is the HIV scheme of the HIV variants. So this HIV is two single-strand RNA virus. The RNA is embedded in a capsid. This is a membrane matrix protein. And this is what we are interested in. The GP120 was a surface protein, glycoprotein, and the transmembrane protein, GP41. So what we are interested in is how it's organized of the GP120 and GP41 as a trimer. So why we are interested in this one? Because this is important for the HIV infected sample. So this is the HIV fusion process. So this is there's a target cell of this virus. So when the virus gets close to the target cell, the CD4, the receptor of the HIV virus on the target cell, bind to the GP120 and make some conformation change of the GP41 and the other envelope spike, and there is a co-receptor, either CCR5 or CXCR4, which is recently dissolved. CXCR4 is recently dissolved by the lab in Professor Liu in colleague. They got this receptor, this GPCRs. That was Professor Liu is very interested in CCR5. So after this one, they get these two membranes get fusion. So from this process, you can see the envelope spike is this protein is very important for the fusion process. So we are interested in how many spikes on the surface and any pattern of the distribution. That's what we can see using EM, and how the component of it, how the trimer, the 3 GP120 and 3 GP41 are organized together, make an envelope spike. So this is the question. So the crystal structure is already solved of the GP120 core, GP41 not really the prefusion study. So what we are really interested in is how the amyloid trimers organization of GP41 and GP110 organized together, and how is the CD4 binding site binding. The question we are addressing this one is basically it's helpful for the vaccine candidate, because GP110 is the most obvious target of the HIV vaccine, because it's the only protein exposed on the surface of the virus and encoded by the virus. So basically, in the vaccine area, they are trying to make the trimer to mimic this trimer on the envelope spike on the surface. So basically, if you can supply the information of the trimer, they can try to mimic this organization, use this as a vaccine candidate. So basically, what we previously used a negative staining of the SIV is kind of another similar to HIV, but even for the monkey. So this SIV, you can see the surface. What we are interested in the envelope spike on the, you can see kind of triangle shape and the distribution. But what we really want is the 3D information, also how it is organized. So basically, we use electron tomography scenes, because we cannot use a single particle because HIV, the virions is various different sizes. So you can it's not homogeneous. So we can, in principle, we cannot use a single particle to reconstruct the HIV virus. So we use focus on the one single virion using electron tomography. That's what we use the single particle electron tomography for the negative staining EM. So this is one single virion. After we got a 3D reconstruction, we can use the section in the computer, the different size. You can see much more clearly this trimer scenes. So people are thinking from biochemical information, they think the GP110 are organized as trimer, but no one see it clearly before. So this is the first picture showing the trimer clearly. Then we can try to fit the GP110 core structure and the glycosylation fitted quite well. But I showed this slides before, but the negative staining also, although you can see it clearly, but the problem is that if you disrupt your virus structure. So basically, the virus after negative stain, for previous slides, after reconstruction of the tomography, the virus is actually squashed like a pancake. So then we trying to switch from negative staining to the cryo-EM, trying to preserve. This is how the cryo-EM put the principle we showed before. Then after we got the sample, we trying to get the tilt series of the electron tomography. This is a virus. So we try to shoot the picture from different angles. That's what electron tomography sees. Then after that, we can trying to get a reconstruction. So you can see from here, this is just one slice of the whole area. You can see the much more clearly the spike compared with our raw images. So the contrast is much better. Instead of besides the envelope spike on the surface, what we are interested in, you can also see the inside, the core of the virus. So this is one of the advantage of the cryo-EM. So if you focus on this virus, this is the whole full section of this single virions. You can see the surface proteins on the top surface and bottom surface and the side views. So I will give you the movie of the. You can see the tomogram different sections. So if we enhance the contrast, you can see much more clearly. So besides the envelope spike, you can also see the core. If we enlarge this one, so this is a spike on this one end, the side surface, and also the other end. So basically, you get the 3D information of the virus. Then this is I didn't spend a lot of time to do the modeling, but this is the idea. So this is get a reconstruction of the whole virus. After you got this one, so we can answer several questions. How many spikes? So this is a what type of SIV, but this is HIV. HIV basically this is different from the previous model, because in the regular model there are up to very regularly distributed envelope spike regular like this one, but actually in wild type HIV there is not only about 14 envelope spike, and also they have a tendency to form a cluster. This is what we found, and this is give some information about because the cluster should be some make the coordinate together to make to get entered into the target cell. That's what something interesting. And also, then we after get this one, we still cannot see a lot of details of the envelope spikes. Then we trying to combine electron tomography and a single particle. So after we got the tomogram of the single virus, but we think the virus, because of different size, we cannot use a single particle analysis. But for the envelope spike, as we treat them as a homogeneous sample, so we can try to cut them into in the volumes into 3D one and try to align them together using a principle of the single particle, trying to push the resolution limit. So that's what we cut them together and try to align, get a 3D averaging of the envelope spike, get a trimer. And this is what we get as a density map. And we can try to fit, that's what we combine different structure, fit the crystal structure of the GP120 core into the density map. And then we try to fit the three GP120 core and try. This is the red one is the CD4 where the receptor binding. So give the information of how CD4 is binding. And also there are some missing loop. That's what cannot be crystallized, be truncated out, so we can see where the missing loop is. But this is still a rough model. So the resolution is beyond is less worse than 3 nanometers. So we cannot see a lot of details here. So trying to after that I came back to IBP. We didn't follow this much, but if we do, actually there are some other people are trying to do this. So you're trying to combine them. For example, after you put the CD4 in, you can see the CD4 binding CD4 here. So this can clarify where the CD4 binding site is. And also there are some antibodies. We know the epitope, so we can see the antibody here. So we can try to identify different locations of the neutralizing antibody binding site. So this is information of the HIV. So basically, the idea, as I mentioned before, the idea, if you want to get the envelope trimer information, the idea is that you want to make as a vaccine to provide supply information for the vaccine design. So basically, there are some company or some lab that we are trying to collaborating. They are making the trimer envelope trimers, but they just using the information, some assay, chemical assays, for example, combined to the widely neutral antibody or something, but they really want to see how their trimer look like. So basically we use single particle, trying to do the reconstruction, get the 3D reconstruction of their vaccine candidate, and then we can structurally combine this with the real envelope trimer, and give their information, oh, okay, you're good enough, or you can change it somehow. So this is what we really can do for the vaccine candidates. So you can structurally combine them together. We have some collaboration with the US lab, so we have some grant on this one. So this is just one example of the electron tomography, and another. Still virus work. So, but this one is a single particle analysis. Just this work is after my lab in IBP was established. So this we got a Cypovirus is a double-strand RNA virus. We got the atomic structure of this virus. This is CPV, we call it Cypovirus. This is a very good model virus for the cryo-EM study. So this is a real virus, the Cypovirus. Basically, this is then we purified a lot of this kind of virus and get the cryo-EM image, we collect a lot of data around 29,000 particles of this image. Then we do use a strategy and principle I introduced in the last session. Do the single particle analysis from this one. We finally get this structure. So this I will skip this slide, but the idea is what I mentioned in the last session before. So we trying to get an initial model and then trying to refine them to get the final reconstruction. I just give you the final results. So this is we collect 29,000 particles and get the initial model and refine them, and finally we get the reconstruction at around 3.9 angstrom resolution. Some professor is expert for this, but actually from this density map, it looks like the side chain is very well. It's maybe I think it's a little bit beyond this. It's better than this resolution. So based on this one, that's what we call the de novo model fitting. So we can try to build the model of the whole virus. So this is a symmetric subunit of the virus. After we build this model, you can apply the symmetry or axial symmetry. You can get the full atomic model of the whole virus. So this is what the protein of the VP1 is a backbone protein of the virus. You can. This is what we built, VP1 and VP1b. And the interesting things after we built the model, we found some interesting things for this virus, because here this domain actually is we identified. This was not identified before, this domain, and this is in the 5-axis pentamer base. People are thinking because this is a double-stranded RNA virus, the RNA is getting out from the center of this channel. But the problem is for this one is that we found there is a spike plugged here. So we really found some interesting is that if you can see this movie, instead of this the center channel, there are several smaller channels here. If you focus on this one, there is a channel, another channel. There are 5 channels surrounding this center area. So this is we think these channels are really the channel of this RNA. The RNA can getting out, and also there are several debates on this one, because there are some information about people. Some people support getting RNA getting out from the centers, but other people think getting out from the peripheral channels. So why the reason we think that the channel is getting out from the small channels, because for the RNA virus, after it capping, for the virus, the RNA must be getting out. Before they get out, they must be capped. This is an important process. After there are several enzymes to capture the mRNA to make the RNA get stable, and they can make new virus. So this after we found this and built this atomic structure, we find there are several enzymes, and actually they are surrounded this channel, small channel. So basically, RNA getting slow, passing by the small channel. They will pass by this enzyme, and this enzyme, sequentially make it capped. So this is why we think the RNA getting out from this virus. So this paper is we submitted and published here. Now we really want to prove this is right. So what we do is we trying to do another stage of this same virus, but here is not wild type, it's transcribing. During the transcription process, we're trying to get the crystal structure of this one. So for this one, this is a channel or model that we support. We think this channel is getting out from here. That's from our work, and also people. but for this transcribing virus is a metastable. It's not very stable, so it's I think not very difficult or even impossible to get it crystallized. So there is no crystal structure reported on any transcribing virus. So people are using cryo-EM. So this is around 2 nanometer resolution transcribing virus. So basically we use, but this is different another virus. So we use the same virus doing a transcribing virus, trying we can make sure it's in the process of transcription by the latest SDS page. And this is non-transcribing, this is transcribing. You can see not much difference. Maybe a little bit. We suspect they are mRNA, but we are not sure. So basically, we use the same technology when you put the transcribing virus in and get the reconstructed this one. We can reconstruct it around 4 angstrom resolution. Recently, we pushed the resolution limit to 3.5 or so. And here you can try to compare these 2 structures of the same virus. The blue one is wild type, and the yellow one is transcribing. So you can see they're almost identical in the backbone area. Only slight difference on this pentamer base, that's where we think the mRNA getting out. So if we can also see some substrate is transcribing the. So we can see this density map cannot be assigned to any backbone protein. So this substrate. So basically, then we can compare. We have the atomic model. Compare this together. So this is this protein. So this protein is VP1. It's a backbone protein. So you can see this non-transcribing a little bit shift to the transcribing stage. But here is this protein. There is another protein, this VP2 tolerant protein. This is where in the pentamer base. So almost the same. You can see this identical here, but here is a little bit. So basically, this is a hinge region. This the whole area move a little bit. So these 2 proteins can change. Only after you get the near atomic resolution, you can see this slight difference. And then we put them in the orientation because you cannot see clearly, get the overview of this. Put it in the orientation of those virus, look like there is opening here. This is if you can see here. So this is wild type, this transcribing, look like they are opening. And here we suspect this is RDRP, but we cannot. We think it's RDRP, but we cannot get the real structure information. So this is they're just like a blower getting open. But if we compare this atomic model together, it look like the virus is a very tiny machine, but a very precise machine. To make it RNA getting out, they have this RNA like a flower blooming, so they are opening and rotating a little bit. After this conformation changes, the RNA is getting out. So this is what we think the model. And then we go back to our question. We really want to prove that this is the channel. So this is wild type. This is transcribing. You can see that the channel is getting open. And also in this here, we can see a little bit density here. We suspect there are RNA, but we cannot, at that resolution, we cannot say it very clear. But now we get more information, look like this is a little RNA. So this is so basically that's what the same story. So we, for the single particle, for this virus, we build the atomic resolution for the transcribing, non-transcribing and the transcribing, we get some information about the different stage. So this is what we really in my lab we are interested in. We are not doing the virus. We want to do the structure of the virus in different stage. For example, transcribing, non-transcribing. Also, we recently get another manuscript accepted was of the adenovirus in different stage. So this is what one direction in my lab. Okay, finally I will give another example of we use both electron tomography and single particle analysis. This is a totally different, it's not a virus, but it's chromatin and epigenetic related. So this is why epigenetic, why we are interested in here. So this is the chromosome, but every people we have 23 pairs of chromosomes. Every cell has the same DNA information, but why they can differentiate to different cells for a neuron cell or some other cells. So there are some controlling it we call epigenetic regulation. So on the process of epigenetic regulation, the chromatin structure plays a very important role there. So for this is a chromosome. So this is the unit of the chromosome is called a nucleosome. Nucleosome is a DNA wrapped around the histone octamer. So the nucleosome then they get in piece of ring. This is a piece of 11 nanometer like this one. 11 nanometer of the nucleosome. Then they fold it into a 30 nanometer chromatin. They fold it together and condense to a chromosome. So that's where around 2 meters length of DNA packed very densely on maybe 20 micrometers. So basically what we are interested in is this one. So how these nucleosomes are organized as into a chromatin. So this is information. This is a textbook. Typically, people think of like this way. So the nucleosome packed like a solenoid model is wrapped this way. But there is still a debate. This is a solenoid model. Or this is this way. But people a lot of information support this kind of zigzag model, the zigzag back. So we need high resolution to see how it's organized. That's what we are thinking. But I will briefly give an intro. So we are working on this one. We try recently. We got a very good reconstruction of the chromatin. We found that very interesting information. We found that chromatin are organized like a DNA is double strand. So this is because still we haven't get it published. So I just give information about it. So we are working on this one and get some interesting results. And also, why is this important for the epigenetic information? Because you can see that the chromosome and the DNA is getting wrapped very condensed very very closely. So all of the information of the epigenetic for the transcription is inactive sometimes. So but why the cell divides into different cells? Some information, for example, here the DNA in this area needed to be open. To make it open, then you transcription active. So there are a lot of mechanisms to make it happen. So for example, some histone modification and the chromatin remodeling composite. There, the cells will recruit some members to make it open. So this is what we call chromatin remodeling complex. There are 4 classes of chromatin remodeling. We are working on this family. So they are by itself. This families they are complex, a big complex. For example, this INO 80. They are about around 16 to 17 subunits. So we are trying to different strategy to work on this one. We're trying to build one by one. This is a typical way. So build it from bottom to top. And also we use E. coli. Some components we use E. coli. Some components we have to use insect cell to express. And also we're trying to make a stable cell line to flag tagged. So we can get grab the whole complex. with the whole component. So I will give you the example. We are still working on this, but we didn't make a lot of progress. But in the process of this one, we find this complex is a subcomplex of the whole complex. We got very interesting. So that complex we call it type 49A and B, or sometimes we call it RVB1, RVB2. It's a core component of a lot of important complexes. It's highly conserved, so we recently get this resolved. This is a family complex. Then we reconstruct this one. This negative stem, we just check the sample and use cryo-EM. So it look like the double. This is a side view and the top view of this one. And we use a single particle analysis, get a reconstruction of this complex. This is A and B. This is a hexamer, double hexamer. And so as I mentioned, so in the cryo-EM field, we got a lot of work on the high residual virus. We can do the near atomic resolution. But this one is, we think is reasonably good, but not only C6 symmetry. It's not a residual symmetry. It's a low symmetry, but we still get this model built. It's a side chain, there's a beta sheet and alpha helix. And also we found this ATP binding, AMPMP is ATP binding, so we can build this model clearly. So for this gives us around 3 point, actually it's better than this one. This is old data. So we now get 3.6 angstrom of this complex. So you can see the side chain here, or even some connection between side chains, the interaction. So this is a very good reconstruction. So basically we got the ATP and the ATP binding state and make them. So we are still we are wrap up this story and trying to make a manuscript on this one. So that's I give several examples of in my lab is virus, chromatin modern complex. So finally, this is I will give the example. So this is we trying to. People are thinking you got the structure of the in vitro. So how about in vivo? So I showed this slide before. So we use correlative microscopy. There's a high resolution. This is even either in crystal structure or the single particle structure. We the atomic resolution. This is light microscopy. So we use electron tomography to bridge the gap, to trying to build, to see how that's what our dream. So how to visualize how these proteins in the cellular background, how they work. So that's what is the platform I mentioned in my previous session. So we use both LM correlative LM with TEM and also SEM. So this is the final movie I want to show. This is that's what I like this movie very much. It's made by Harvard University. So you can see so you can see here the song. This is but I'm not very familiar because my background is not biology background actually, but I can know a little bit. But this you can see a lot of materials they used to make the movie. For example, this one by cryo-EM. So this is how it works. This is like a B cell, how it works. The ribosome, a lot of things. This is the final movie and the final of the last slide of my talk. So we can I think maybe a lot of you have already seen this movie, but this is I think it's very elegant. I think a lot of this is made by Hongwei Wang. this part. This is a category, so You can see this one, a lot of documents are made by EM fields. Yeah. So we hold one, but this is a gym. This is they just put a lot of things together to make this ribosome. Yeah. So I will. Yeah, this is now this is our institute in the Olympic Park area in Beijing. This is where Professor Liu and I was in. So we are in this building actually. Our scope is here. In this building. So finally, this is my lab. Thank you very much.
Thank you.